You know, I was looking through the prep for today, and this one organ kept coming up with these, well, frankly, incredible descriptions. Things like, it can regenerate, it's a master chemist, a gatekeeper, even a waste disposal unit. It almost sounds like biologically impossible.
It does sound pretty amazing when you list it like that.
So what are we actually talking about today? Okay, let's unpack this. Today we are taking a deep dive into the liver. Truly one of the body's most dynamic and frankly essential organs. The complexity is just staggering.
It really is.
And our mission today is to really peel back the layers, focusing on its transport and metabolic roles. We're aiming to use expert sources to understand its functions, almost like, elite professionals in the field,
Yeah.
getting into the intricate structures, the crucial processes, and those clinical insights that really drive home its importance.
What's fascinating here is just how um, fundamental the liver is. It's not just sitting there. It's a genuine metabolic powerhouse. It's deeply connected with the whole GI system.
Right.
And it's responsible for this just astonishing range of jobs, from processing pretty much every nutrient you absorb to neutralizing and getting rid of all sorts of toxins. It's really at the heart of so much that keeps us going.
So let's start with the basics, just getting our bearings. Where exactly is the superstar organ located?
Okay, so you'll find it's a large organ, has multiple lobes nestled right up there in your upper right abdominal cavity, kind of tucked under your diaphragm,
Okay.
and it works very, very closely with your entire digestive system. Its main functions really tell the story. Think of it as the body's central hub, maybe.
A hub, yeah.
Yeah.
Receiving and distribution.
Exactly. First, it's the initial processing site for most nutrients absorbed from your food. Second, it's key for digestion itself because it makes and secretes bile acids, absolutely critical for handling fats.
Bile acids, right. We'll get more into those.
We will. Third, and this is huge, it is that metabolic powerhouse that deals with metabolic waste products and also xenobiotics.
Xenobiotics, so foreign things like drugs or chemicals.
Precisely. Things from outside the body. The liver processes them, converting them so we can excrete them. And finally, it stores and produces a whole bunch of essential stuff. Glucose for energy, amino acids, crucial plasma protein.
Oh, so these functions, we can sort of group them, right? The source material mentions 3 key areas.
Yeah, broadly speaking, its contributions to whole body metabolism, its role in detoxification, and then its unique ability to excrete certain waste products, especially ones bound to proteins or fats that the kidneys struggle with.
Okay, let's dig into that first one. The liver as a metabolic mastermind. We're talking about the main cells, hepatocytes, right? And how they handle carbs, fats, proteins. How do they juggle all that?
It's pretty remarkable. Let's start with carbohydrate metabolism. The liver is absolutely pivotal for keeping your blood sugar stable.
Right, glucose levels.
Exactly. One key process is gluconeogenesis, literally making new glucose. The liver can actually convert things like amino acids or even parts of fats into glucose.
Why is that so important?
Well, it's how your body keeps blood glucose levels steady between meals.
Mm-hmm.
It makes sure your brain, especially, has a constant fuel supply, even if you haven't eaten for hours.
Ah, okay. So if the liver isn't working well,
Then you can run into problems.
yeah.
You might see dangerously low blood sugar, hypoglycemia. It shows just how vital that function is.
Okay, so that's carbs. What about lipids, fats?
Right, lipid metabolism. Hepatocytes are just packed with enzymes for breaking down fatty acids. That's fatty acid oxidation, which provides a lot of energy for the body.
Okay.
And, kind of interestingly, they can also take products from carbohydrate metabolism and convert them into lipids. Those then get packaged up and sent off for storage in adipose tissue, fat tissue.
So it makes fat too.
It does. And it also synthesizes large amounts of other essential fatty things. Lipoproteins, which are like transport vehicles for fats in the blood, cholesterol, and phospholipids. And a good chunk of that cholesterol it makes gets converted into bile acids.
Which loops back to digestion again.
Exactly. It's all very interconnected.
And the third group, proteins.
Yes, protein metabolism. This is another huge area. The liver makes many of the non-essential amino acids, the ones your body can build itself.
Okay.
It also does clever things like interconverting amino acids or deaminating them.
Yeah.
Basically stripping off the nitrogen part so the leftover carbon structure can be used to make carbs or fats.
Resourceful.
Very. And here's a big one. Apart from antibodies made by your immune system, the liver synthesizes almost all the proteins circulating in your blood plasma.
Really? Like what?
Well, the most famous is probably albumin. Albumin is critical for maintaining plasma oncotic pressure. essentially. It keeps fluid inside your blood vessels.
Ah, so it stops you from getting leaky or swollen.
Exactly. And it also makes most of the clotting factors needed for blood coagulation.
Okay, so that must have clinical implications if the liver's damaged.
Absolutely. Clinically, this is really important. Patients with advanced liver disease often develop peripheral edema, swelling in the limbs because their albumin levels are low.
Right, the fluid leaks out.
Yep. And they're also prone to bleeding disorders because they can't make enough clotting factors. And one more crucial protein-related job, ammonia disposal. Ammonia is a toxic byproduct of protein breakdown,
Toxic, how so?
especially to the brain. The liver takes this toxic ammonia and converts it into a much safer compound called urea, which the kidneys can then easily filter out and excrete. It's a critical detoxification step.
Wow, okay, that leads perfectly into detoxification. It sounds like the liver is the body's ultimate gatekeeper and garbage disposal.
Yeah.
How does it manage that? What's the secret?
A huge part of it is its unique blood supply. It's quite different from other organs. Most organs get oxygenated arterial blood, right?
Right.
The liver gets that too. But the majority of its blood supply, we're talking over 70% when you're resting, maybe over 90% after a meal, it's actually venous blood.
Venous blood from where?
It comes directly from your intestine via the portal vein.
Ah, so everything absorbed from the gut goes straight to the liver first.
Pretty much. Nutrients, yes, but also potential or harmful stuff you might absorb, drugs, toxins, bacterial bits. This strategic location is key.
That explains the first pass effect with some drugs, doesn't it? Where the liver chews them up before they even get into general circulation.
Exactly that. Some oral meds just don't reach effective levels because the liver metabolizes so much on that first pass.
So how does it actually defend against all this incoming stuff?
It has two main levels of defense. First is physical. As blood flows through these special liver blood vessels, the sinusoids, it encounters specialized immune cells called Kupffer cells.
Kupffer cells, like guards?
Sort of like tiny, very effective vacuum cleaners, maybe. They're macrophages, so they literally engulf particulate stuff, bacteria that might sneak in from the colon, other debris. They just gobble it up.
Okay, physical filtration, what's the second level?
That's biochemical. The hepatocytes themselves are loaded with a huge arsenal of enzymes. Their job is to modify toxins, both ones we make ourselves, endogenous, and ones from outside, exogenous.
Modify how?
The main goal is to make them more water-soluble. This makes them less likely to be reabsorbed later in the intestine and much easier for the body to excrete, either in bile or urine.
Okay, makes sense. Easier to flush out.
Precisely. And these reactions are often divided into two phases. Phase I reactions usually involve enzymes adding or exposing small chemical groups on the toxin, kind of like preparing it for the next step. A famous family of enzymes involved here is the cytochrome P450 system.
Cytochrome P450, yeah, hear that a lot with drug metabolism.
Exactly. Then comes phase II reactions. Here, the modified toxin is conjugated. Basically, it's attached to another, usually larger, water-loving molecule, things like glucuronic acid, sulfate, or certain amino acids.
So phase I prepares it, phase II tags it for disposal.
That's a great way to put it. This tagging, or conjugation, makes it much more water-soluble and flags it for export. These modified tagged products are then either shunted into bile for excretion via feces or sent back into the bloodstream for the kidneys to filter out in urine. It's a really sophisticated two-step chemical processing line.
Incredible. And you mentioned excretion via bile specifically, for stuff kidneys can't handle easily.
Yes, exactly. Things that are tightly bound to proteins in the blood or are very lipid soluble are hard for kidneys to filter. The liver handles these by actively pumping them into bile. Hepatocytes have specific transporters, like molecular gates, on their surface facing the blood.
To bring things in.
Right. High-affinity transporters grab these substances from the blood. Then, after maybe being modified in phase I or II, other specific transporters on the opposite side of the cell, the side forming the bile channel, pump them out into the bile.
So specific pumps for import and export?
Absolutely. We know this partly from genetic conditions where one of these pumps is faulty, causing major problems. For example, there's a pump called MRP2 that's crucial for getting conjugated bilirubin into bile. Anyway, once in the bile, its unique detergent-like properties help keep even fatty waste products dissolved, allowing them to be carried out into the intestine and eventually eliminated. It's very targeted transport.
Here's where it gets really interesting for me. We've talked functions, metabolism, detox, but how does the liver physically do all this? Its structure, its architecture must be key, right?
Oh, absolutely. The design is critical. The main cells, the hepatocytes, aren't just randomly scattered. They're arranged in these interconnected cords or plates. Imagine like intricate walls forming a maze.
Okay, a maze of cells.
And blood flows through this maze, bathing the cells. The liver gets a massive blood flow, way more than you'd expect for its size. This ensures plenty of oxygen and nutrients for all that metabolic work. And as we said, most of it, especially after eating, is that nutrient and toxin-rich portal vein blood.
So how does the blood flow through this maze?
The plates of hepatocytes are supplied by these unique, low-resistance blood vessels called sinusoids. They get blood from both the portal vein branches and the hepatic artery branches.
Sinusoids. How are they different from normal capillaries?
Well, for one thing, their resistance is very low. Also, many can be sort of collapsed when you're fasting, but then they open up or get recruited when blood flow increases after a meal. This lets the liver handle that big surge of blood without the pressure skyrocketing.
Smart design.
Very. And the cells lining these sinusoids, the endothelial cells, are also special. They have these large pores or openings called fenestrations. Think of them like tiny windows.
Windows. Big enough for what?
Big enough for large molecules like albumin, the main protein in blood, to pass right through. Plus, they don't have a continuous basement membrane underneath them like most capillaries.
So, less of a barrier.
Exactly. No real diffusion barrier. This means albumin and anything bound to it has easy access to the surface of the hepatocytes for uptake. And nestled among these endothelial cells are those Kupffer cells we mentioned, the immune guards.
Okay, so blood flows through leaky sinusoids, past Kupffer cells, right next to the hepatocytes.
Precisely. And between the sinusoid lining and the hepatocytes, there's a very thin layer of loose connective tissue called the space of Dis. Normally, it offers almost no resistance to molecules moving between the blood and the liver cells.
So everything gets easy access.
Pretty much. And in this space of Dis, you find another really important cell type, stellate cells.
Stellate cells, what do they do?
Normally, they're involved in storing retinoids related to vitamin A, and they produce growth factors for hepatocytes. But, and this is clinically crucial under conditions of chronic injury, like from alcohol or viruses.
Uh-oh.
Yeah. These stellate cells get activated, and when they're activated, they start churning out huge amounts of collagen.
Collagen. Like scar tissue.
Exactly like scar tissue. This fibrosis is a major contributor to progressive liver dysfunction.
Okay, one more structural thing, these zones. How does that work?
Right, the liver lobule, the functional unit, is often described in terms of zones based on blood flow. You have branches of the portal vein, hepatic artery, and bile duct running together. That's the portal triad, usually at the periphery of the lobule.
Okay.
Cells closest to this triad are called periportal, or zone 1. They get the blood with the highest oxygen and nutrient levels first. They're typically most active in detoxifying things under normal conditions, but may be more vulnerable to certain types of oxidative injury.
And the other end?
Cells furthest from the triad and closest to the central vein where blood drains out are pericentral, or zone 3. They get blood that's already passed through zones 1 and 2, so it's lower in oxygen. These cells are more sensitive to ischemia, lack of oxygen. Interestingly, they seem to be more involved in things like bile acid synthesis.
And zone 2 is just in between?
Yep. Zone 2 is intermediate.
Okay.
What's clinically useful is that the location of damaged cells on a liver biopsy can give doctors clues about the type of injury. Zone 3 damage might suggest ischemia or certain toxins, while Zone 1 damage might point to others.
That is incredibly detailed architecture. It makes sense now how structure enables function, but it also sounds delicate. How do problems with this structure lead to serious clinical conditions?
Yeah, this is where the basic science connects directly to patient problems. Let's talk fibrosis and cirrhosis. When scar tissue, that collagen laid down by activated stellate cells, starts building up, it compresses those sinusoids.
Squeezes the blood vessels.
Right. The liver loses that ability to handle increases in blood flow, especially after meals, without the pressure going up. And remember those leaky fenestrations. With higher pressure, plasma fluid, rich in albumin, starts to leak out more readily.
Into the abdomen.
Yes. It overwhelms the lymphatic system's ability to drain it, and fluid accumulates in the abdominal cavity, that's the sites. Causes that characteristic abdominal swelling you see in advanced liver disease.
And the pressure buildup itself.
That's called portal hypertension. The pressure in the portal vein system gets dangerously high. The body tries to adapt by forming new blood vessels, collaterals, to bypass the blockage in the liver.
Bypassing the liver? That doesn't sound good.
It's not ideal. Some of these collaterals form in the esophageal varices. They have thin walls, they're under high pressure, and they can rupture, causing catastrophic bleeding. A true medical emergency.
Wow.
And even if they don't bleed, these bypass vessels mean that blood from the gut, potentially carrying toxins like ammonia, doesn't get filtered by the remaining healthy liver tissue. So toxin levels rise in the general circulation, affecting other organs, especially the brain.
And cirrhosis is when this scarring gets really bad.
Exactly. Cirrhosis is the end stage of chronic liver injury from viruses, alcohol, other causes. The constant damage kills hepatocytes, activates those stellate cells, and leads to widespread, irreversible fibrosis. Scar tissue replaces functional liver tissue, shrinking the liver's capacity to do everything: synthesis, metabolism, excretion. Eventually, for end-stage liver disease, transplantation is often the only option.
Such a devastating consequence of that structural damage. What about hepatitis? That's inflammation, right?
Right. Hepatitis means inflammation and necrosis, cell death, in the liver. There are 5 main viruses known to cause it: hepatitis A through E.
Okay. Are they all the same?
No, they differ significantly. Hepatitis A and E are usually caught from contaminated food or water, fecal oral route. They typically cause acute, self-limited infections. You get sick, then you recover.
Okay, those sound less scary.
Generally, yes.
Yeah.
But hepatitis B and C are different. They're usually transmitted through blood or body fluids parenterally, and they, along with hepatitis D, which needs hep B to survive, often lead to chronic infection.
Chronic, meaning long-term problems.
Yes, chronic HBV and HCV significantly increase the risk of developing cirrhosis and liver cancer, hepatocellular carcinoma. For years, the outlook for people with chronic HBV or HCV was pretty grim. HBV is still a leading cause of liver-related deaths globally, and HCV was the top reason for liver transplant.
Has that changed?
Dramatically. It's been a real revolution in medicine. We now have an effective vaccine for hepatitis B, and antiviral drugs can control the infection really well.
That's great. And hep C?
Even better, in a way. Hepatitis C is now largely curable. New oral antiviral medications directly target the virus's replication machinery. They have incredibly high cure rates. It's completely transformed the landscape for HCV patients. Truly a game changer.
That's fantastic news. Real progress. Okay, so we've covered cells, structure, metabolism, detox, clinical issues. Let's shift focus now to bile. What exactly is this stuff, and how is it made and handled?
Okay, bile. It's essentially the liver's excretory fluid, but it's also vital for fat digestion. It starts being formed right inside the hepatocytes. Remember how they're arranged in plates?
Yeah, the maze walls.
Well, the apical surfaces, sort of the top sides of adjacent hepatocytes, face each other and form these tiny channels called canaliculi. These are the starting point for the bile drainage system.
Little tubes between the cells?
Exactly. And hepatocytes have specialized pumps on this apical membrane that actively transport solutes into these canaliculi. That's what initially forms bile.
What solutes? What's in it?
The key components are bile acids, phosphatidylcholine, which is a type of phospholipid, a fat, and cholesterol. These are all dissolved together in a watery solution, forming what we call a micellar solution. Think of micelles as tiny little clusters or aggregates of these fatty molecules.
Okay, bile acids, fats, cholesterol, water.
Right. And the active pumping of these solutes into the canaliculus is what drives the initial bile formation. This canalicular bile then flows out and gets modified further down the line. Its main purposes are to help dissolve fats in the intestine so we can absorb them, and to carry waste products out of the body.
Where does it go after the canaliculi?
These tiny channels drain into slightly larger biliary ductules. These are lined by different cells called cholangiocytes, more typical epithelial cells. These ductules then merge into larger bile ducts, eventually forming the main right and left hepatic ducts that carry bile out of the liver. These join to form the common hepatic duct. From there, bile can either flow into the gallbladder for storage via the cystic duct, or go straight down the common bile duct towards the intestine, depending on whether the exit sphincter is open or closed.
It sounds like a plumbing system.
It really is an intricate plumbing system. And we know how critical each part is because of rare genetic conditions. There are syndromes called progressive familial intrahepatic cholestasis, PFIC, where mutations in specific transporter pumps cause severe liver disease because bile can't flow properly or has the wrong composition.
Like if a pump is broken.
Exactly. For example, in PFIC type 2, the pump for bile acids themselves, called BSE, is faulty, so hardly any bile acids get into the bile. In PFIC type 3, a different pump, MBR3, that moves phosphatidylcholine into bile is broken. Without phosphatidylcholine, the bile acids become much more damaging to the duct lining cells. It really highlights how essential each specific molecular part is.
That cellular detail really drives it home. Let's zero in on those core components, the bile acids. How are they made, And what's this enterohepatic circulation thing?
Bile acids are really fascinating. They're made by hepatocytes directly from cholesterol. It's one of the main ways the body gets rid of excess cholesterol.
Okay, so cholesterol is the starting material.
Yes, there's a key enzyme, the rate-limiting step, that modifies cholesterol to start the process. This eventually yields the primary bile acids, mainly chenodeoxycholic acid and cholic acid.
And is this production constant?
No, it's tightly regulated. The liver adjusts synthesis based on need. If the amount of bile acids returning to the liver in the portal blood drops, synthesis can ramp up significantly, maybe tenfold.
How does the liver know?
The returning bile acids themselves act as signals. They activate receptors on or inside the hepatocytes, which then control the genes for the synthesizing enzymes. It's a feedback loop. If bile acid levels are high, synthesis is suppressed.
Smart. So primary bile acids are made in the liver. What about secondary ones?
Ah, good point. Once primary bile acids reach the large intestine, the bacteria living there can modify them. Bacterial enzymes convert primary bile acids into secondary bile acids, like deoxycholic acid and lithocholic acid.
So bacteria play a role too.
They do. Now, whether primary or secondary, before leaving the liver cell, almost all bile acids undergo another crucial step: conjugation.
Conjugation again, like with the toxins.
Similar idea, different molecules involved. Here, the bile acids are attached to either glycine or taurine. This conjugation dramatically changes their chemical properties, specifically lowering their pKa.
pKa, that affects how acidic they are?
Sort of. It makes them much more likely to be ionized, to carry a negative charge at the pH found in the small intestine. And this is key.
Why?
Because charged molecules can't easily slip across cell membranes passively. So, conjugated bile acids stay trapped in the intestinal lumen where they're needed to help digest fats. They can only be reabsorbed later, way down in the terminal ileum, by a specific active transporter protein called ASBT.
Okay, so conjugation keeps them where they need to be.
Exactly. If any bile acids escape that active uptake and reach the colon, bacteria there can deconjugate them, remove the glycine or taurine. These unconjugated bile acids are less charged and can be passively reabsorbed across the colon wall.
It seems complicated. Why all this back and forth?
It all ties into the enterohepatic circulation. This is the liver's amazing recycling system for bile acids. You need a large amount of bile acids in your gut to digest a fatty meal effectively, but your body doesn't want to have to make that much from scratch every single time.
So it recycles them.
Massively. Those conjugated bile acids actively reabsorbed in the ileum travel via the portal vein straight back to the liver. Hepatocytes are incredibly efficient at grabbing them back out of the blood using specific transporters on their surface.
Okay.
And even the unconjugated ones absorbed from the colon return to the liver, where they get reconjugated and sent out again. The whole pool of bile acids circulates multiple times during a single meal.
Wow. How efficient is it?
Extremely. Only a small fraction, maybe 5–10% of the total pool, escapes reabsorption and is lost in the feces each day. The liver just needs to synthesize enough new bile acids daily to replace that small loss.
That's incredible conservation.
It is, and it links directly back to cholesterol. Remember, bile acids are made from cholesterol, so the body's cholesterol balance depends on synthesis versus loss, and bile is the main route for cholesterol loss, either as cholesterol itself or as bile acids.
Ah, so even with recycling, making bile acids is a way to lose cholesterol.
Yes. Even though most are recycled, that small daily loss that needs replacing accounts for a significant chunk of daily cholesterol elimination. This is why some cholesterol-lowering strategies work by interrupting the enterohepatic circulation. If you block bile acid reabsorption, the liver has to pull more cholesterol from the blood to make new bile acids, lowering overall cholesterol levels.
Clever therapeutic angle. So bile acids are central, but what else is in bile and how does it get fine-tuned?
Right, it's not just bile acids. We mentioned cholesterol and phosphatidylcholine also get transported into the bile by specific pumps on the hepatocyte membrane. ABCG 5G8 for cholesterol, MDR3 for phosphatidylcholine. Okay. These, along with the bile acids, form those mixed micelles. Because these micelles are osmotically active, they draw water flows into the canaliculi too, along with other small solutes from plasma like calcium, glucose, amino acids, urea. They just follow the water, essentially.
So the initial bile is kind of like plasma with added bile components?
Roughly, yes. Plus you have conjugated bilirubin being pumped in and various other metabolic waste products and processed xenobiotics.
And then it gets modified. You mentioned the ductules.
Exactly. As this primary bile flows through the biliary ductules lined by cholangiocytes, it gets fine-tuned. These cells actively reclaim useful things like glucose and amino acids, pulling them back out of the bile so they're not wasted.
Okay, reclaiming the good stuff.
Right. They also modify the electrolytes. They exchange chloride ions from the bile for bicarbonate ions, pumping bicarbonate in. This makes the bile slightly alkaline.
Why alkaline?
It helps keep calcium dissolved, reducing the risk of calcium salts precipitating out, which could form stones. These cells also have enzymes on their surface, like GGT, that break down other molecules like glutathione, allowing the pieces to be reabsorbed.
DGT, that was one of the tests you mentioned for biliary injury.
It was. High levels suggest cholangiocyte damage. Finally, these ductual cells can also dilute the bile, especially after a meal. Hormones like secretin stimulate them to secrete more bicarbonate and water, increasing overall bile flow precisely when it's needed most for digestion. It's a dynamic process.
Constantly adjusting. Okay, after all that, the bile needs somewhere to go between meals. Enter the gallbladder, the storage tank. Often ignored until it causes pain, right?
Pretty much, yeah. The gallbladder's main job is storage and concentration. Between meals, a muscle called the sphincter of Oddi at the end of the common bile duct constricts, closing off the direct path to the intestine. So bile backs up and flows into the gallbladder via the cystic duct.
And it just sits there?
Not just sits there, it gets concentrated. The gallbladder lining is made of specialized epithelial cells that actively pump sodium ions out of the bile. Water follows the sodium, so the bile becomes much more concentrated.
How much more concentrated?
The concentration of the main solutes, bile acids, cholesterol, phospholipids, can increase by more than tenfold.
Wow. Doesn't that cause problems? Like, wouldn't it draw water back in osmotically?
That's the clever part. Even though the concentration of individual molecules goes way up, the osmotic activity doesn't change much. Why? Because these molecules cluster together in those micelles. A whole micelle acts as just one osmotic particle. So as water leaves, any free monomers just join existing micelles. Bile stays roughly acetonic with plasma.
Neat trick. Does concentrating help?
It reduces the volume needed for storage, and it might also help keep cholesterol dissolved by keeping the bile acid concentration high relative to cholesterol. But gallstones. Humans seem particularly prone to them. They're precipitates, usually of cholesterol, or sometimes calcium bilirubinate.
What problems do they cause?
The main issue is obstruction. If a stone blocks the cystic duct, or the common bile duct, it causes pain, biliary colic, especially after fatty meals. It can lead to inflammation, poor fat digestion, and if the blockage persists, bile constituents back up, potentially causing jaundice and liver damage.
Why do they form? Is the bile just too concentrated?
Cholesterol saturation is part of it. Many people actually have bile that's supersaturated with cholesterol, but normally other proteins in bile inhibit crystal formation or nucleation. However, if bile sits for a long time in the gallbladder, the chances of nucleation increase. Hence the maybe slightly anecdotal advice about not skipping meals to keep the gallbladder emptying.
Interesting. Are some people more at risk?
Yes, the classic risk factors are female, middle-aged, obesity, and having had children. Estrogen seems to increase hepatic cholesterol secretion into bile, tipping the balance towards precipitation.
And treatment.
Often surgical removal of the gallbladder, usually done laparoscopically these days. Small stones stuck in the ducts can sometimes be removed with an endoscope.
Okay, so the gallbladder stores and concentrates. How does it release the bile when needed?
When you eat, especially fat, signals are sent that cause the sphincter of Oddi to relax, open the gate to the intestine, and simultaneously cause the smooth muscle in the gallbladder wall to contract strongly.
Squeezing the bile out.
Exactly. The key hormonal signal for this is calcicekinin, CCK, released from the intestine in response to fats and amino acids. Nerve pathways, both local reflexes and signals via the vagus nerve, some triggered by CCK itself, also contribute. The result is a nice coordinated ejection of that concentrated bile right into the duodenum, ready to help with fat digestion.
Such coordination. Okay, moving from bile storage to a specific component. Bilirubin, the stuff that causes jaundice and colors. Other things.
Yes, bilirubin. It's the breakdown product of heme, the iron-containing part of hemoglobin from old red blood cells mostly. It's potentially quite toxic.
Toxic.
Yes, especially the initial form, unconjugated bilirubin. If levels get too high, it can cross the blood-brain barrier and damage brain cells. This is a major concern in newborns with severe jaundice. But I might also have some antioxidant properties, and it's definitely the body's way to get rid of excess heme.
Where does most heme come from?
About 80% is from the hemoglobin, senescent old red blood cells being broken down. The rest comes from other heme-containing proteins in the body. And yes, bilirubin and its downstream metabolites give bile its color, feces its characteristic brown color, and contribute to the yellow color of urine.
And jaundice is just too much bilirubin in the blood.
Exactly. Accumulation in the circulation leads to it depositing in tissues, causing that yellowish discoloration of the skin and the whites of the eyes.
How is it actually...
It's a two-step process, mainly happening in phagocytic cells, like macrophages in the spleen and the Kupffer cells in the liver. First, an enzyme called heme oxygenase breaks open the heme ring, releasing iron and producing a green pigment called biliverdin.
Green? I thought bilirubin was yellow.
It is. A second enzyme, biliverdin reductase, quickly converts the green biliverdin into the yellow pigment, bilirubin. This initial bilirubin is not water-soluble, so it hitches a ride on albumin protein to travel through the bloodstream.
Okay, so it gets made, travels on albumin. Then what?
Then it gets to the liver. Hepatocytes take it up from the blood, using specific transporters like OATPs. Inside the hepatocyte, it undergoes that crucial conjugation step we mentioned earlier.
Adding glucuronic acid.
Yes, usually 1 or 2 molecules of glucuronic acid are attached by the enzyme UDP glucuronal transferase. This makes the bilirubin water-soluble.
Why is UGT important again? Newborns?
Right. UGT activity is low at birth and takes a little while to ramp up. That's why mild jaundice is common in newborns. They're a bit slow at conjugating the bilirubin initially.
Okay, so now it's conjugated, water-soluble.
Now, it can be actively pumped out of the hepatocyte into the bile canalicula's. This uses that specific transporter pump we mentioned, MRP2. Once it's in the bile as conjugated bilirubin, it generally can't be reabsorbed from the intestine, so it's destined for excretion.
Is the system perfect? Does all of it get excreted immediately?
Not quite. The transport isn't 100% efficient, so there's normally a small amount of both unconjugated and conjugated bilirubin circulating in the plasma, bound to albumin. Conjugated bilirubin is bound less tightly, though, so if levels rise, it can get filtered by the kidneys and appear in the urine, making it dark.
What happens to it once it reaches the colon and bile?
Gut bacteria get to work again. They first deconjugate it, removing the glucuronic acid. Then they metabolize the bilirubin further into colorless compounds called urobilinogens. Some urobilinogen is reabsorbed back into the blood, circulates, and can be either re-excreted by the liver or filtered by the kidneys into urine. The rest is converted by bacteria in the colon into colored compounds like urobilins and stercobilins, which are finally excreted in the feces and give it its color.
So measuring bilirubin in blood is clinically useful.
Very. Distinguishing between unconjugated and conjugated forms helps diagnose problems. High unconjugated bilirubin suggests either the liver isn't conjugating it properly, like in newborns or genetic UGT defects, or there's a massive overload of heme breakdown, like after a bad blood transfusion reaction, overwhelming the system. Unconjugated bilirubin won't show up in urine because it's stuck to albumin.
And high conjugated bilirubin.
That usually means bilirubin is being conjugated okay, but it can't get out into the bile properly. Maybe there's a blockage, or a problem with the MRP2 export pump. Since conjugated bilirubin is water-soluble and less tightly bound to albumin, it will spill over into the urine, making it dark.
You mentioned genetic defects, like Crigler-Najjar.
Exactly. That's a perfect example of how vital that UGT enzyme is. Crigler-Najjar syndrome is caused by mutations in the UGT gene. Type I is the severe form, complete lack of the enzyme. Unconjugated bilirubin builds up to dangerous levels, causing brain damage, kernicterus. Liver transplant is often the only cure, though gene therapy is being explored.
And type 2?
Type 2 is milder, some residual enzyme activity, maybe 10%. Jaundice is less severe. It can sometimes be managed with things like phototherapy using blue light, helps convert unconjugated bilirubin in the skin into water-soluble forms that can be excreted without needing conjugation. It just highlights the critical role of that single enzyme step.
Amazing how one enzyme can be so critical. Okay, last major topic, ammonia. Sounds simple, NH3, but you said it's really toxic.
Extremely toxic, especially to the central nervous system. Ammonia is a small, neutral molecule, so it crosses cell membranes very easily. It's produced mainly from protein breakdown, both in our tissues and by bacteria in the gut.
And the liver deals with it.
The liver is absolutely essential for keeping ammonia levels safe. It does this by converting ammonia into urea using a pathway called the urea cycle, sometimes known as the Krebs-Henselite cycle.
Urea. That's much less toxic.
Much less toxic and easily handled by the kidneys. And here's the key thing. The liver is the only tissue in the entire body equipped with the full set of enzymes needed for the urea cycle.
Only the liver can make urea.
Correct. That makes its role in ammonia detoxification completely unique and indispensable.
Where does all this ammonia come from?
About half of it originates in the colon, produced by bacterial enzymes breaking down urea or amino acids. Some is absorbed from there. Another significant chunk, maybe 40%, comes from the kidneys as part of their own metabolic processes. The remaining 10% or so comes from amino acid breakdown in the liver itself, muscle metabolism, and other minor sources.
How does the liver keep levels low?
It's incredibly efficient at pulling ammonia out of the blood, arriving from both the portal vein, carrying the gut-derived ammonia, and the general circulation. Because ammonia is so permeable, it readily enters hepatocytes.
And then straight into the urea cycle.
Yep, it enters the cycle, gets converted to urea. The urea then leaves the hepatocyte, goes back into the bloodstream, travels to the kidneys, and gets filtered out into urine. About half the urea filtered by the kidneys is excreted.
What happens to the other half?
Some diffuses back into the blood, and some urea actually finds its way into the colon, where bacteria can break it back down into ammonia again.
Wait, it gets recycled back into ammonia.
Some of it does, yeah. That ammonia is then either reabsorbed and heads back to the liver, or gets trapped and excreted in feces. It's a complex balance, but the net effect in health is efficient ammonia removal via urea synthesis.
So what happens if the liver fails, clinically?
It's devastating. In acute liver failure, where metabolic capacity crashes suddenly, ammonia levels can rise very rapidly, leading quickly to coma and death due to brain toxicity.
And in chronic liver disease.
But often a more gradual decline. As liver function deteriorates over time, ammonia and other toxins that the liver normally clears start to build up. This leads to a condition called hepatic encephalopathy.
Encephalopathy, brain problem.
Yes. Symptoms can range from subtle changes in personality or sleep patterns to confusion, memory loss, poor coordination, and eventually profound drowsiness and coma. It's a major complication of advanced liver disease and can be fatal if not managed. It really underscores how vital that ammonia detoxification role is.
It truly does. Okay, we've covered an immense amount about this incredible organ. To wrap things up, how do doctors actually assess liver health in practice? What are the key diagnostic tools?
Right, the liver function tests, or LFTs, although not all strictly measure function. They have several goals. Assessing if hepatocytes are injured, checking if bile excretion is blocked, evaluating the health of the bile duct cells, cholangiocytes, and monitoring treatment or transplant status.
So how do they check for hepatocyte injury, dead liver cells?
When hepatocytes are damaged or die, say, from inflammation or infection, they leak enzymes into the bloodstream. Two key ones we measure are ALT, alanine aminotransferase, and AST, aspartate aminotransferase.
ALT and AST, those are common tests.
Very common. Elevated levels in the blood are a direct indicator of liver cell injury. ALT is generally considered more specific to the liver, as AST is also found in muscle, including the heart.
Okay, so all tests for liver cell damage. What about problems with the bile ducts?
For that, we look at different markers. Alkaline phosphatase, ALPHOS or ALP, is an enzyme found on the bile canalicular membrane of hepatocytes. If bile flow is blocked somewhere, ALP levels often rise in the blood. Another marker is GGT, gamma-glutamyl transpeptidase, which we mentioned is found on cholangiocytes. Elevated GGT suggests damage specifically to the bile duct lining cells.
So different tests point to different types of problems.
Exactly. And then there are tests that reflect the liver's overall synthetic or metabolic function.
Like making proteins.
Yes. Measuring serum albumin levels tells you how well the liver is making this crucial protein. Low albumin can indicate chronic liver disease. Another key one is measuring blood clotting time, often via the prothrombin time, PT or INR. Since the liver makes most clotting factors, a prolonged clotting time suggests impaired synthetic function.
And bilirubin.
Measuring bilirubin levels, both total and distinguishing conjugated from unconjugated, gives crucial information about the liver's ability to process and excrete this waste product, as we discussed.
So you put all these blood tests together.
Yes, the pattern of abnormalities across these tests, combined with the patient's symptoms and history, helps pinpoint the nature and severity of liver disease. Plus, in patients with known liver disease, monitoring blood glucose and ammonia levels is often important too, given the liver's role there.
And it's not just blood tests, right?
No, absolutely not. Imaging studies like ultrasound, CT scans, MRI are vital for looking at the liver structure, detecting masses, assessing blood flow, and checking the bile ducts. And sometimes, a liver biopsy, examining a tiny piece of liver tissue under a microscope, is needed for a definitive diagnosis or to stage the extent of damage, especially fibrosis.
Wow. An incredible array of tools to investigate this complex organ. We've certainly taken a profound journey today through the liver's metabolic genius, its detox systems, its role in digestion and excretion. It really is one of the body's most pivotal, hard-working organs.
It truly is.
Yeah.
And we connect this to the bigger picture. You see how interconnected everything is.
Mhm.
Understanding the transporters helps understand bile formation, which helps understand digestion, which links back to metabolism. Damage to structure impacts function across the board. Appreciating any one part really deepens your respect for the whole system.
Absolutely. And it's amazing how much we continue to learn. These deep dives are so valuable for really getting to grips with these complex topics, aiming for that professional level of understanding. So what does this all mean for you listening? Well, here's a thought to maybe mull over. Considering the liver's incredible resilience, its capacity for regeneration, but also its vulnerability to chronic damage and its absolutely vital roles.
Oh.
What are the broader societal and public health implications of truly understanding and, most importantly, protecting such a crucial organ?